KURINJI Senior Secondary School, CBSE, Namakkal.
Successful result producing school in state every year.
Administered by the Board of Directors with over 35 years of experience in the teaching field.
Special attention on art, craft, music, dance, silambam, yoga, skating and karate.
Focus on Hindi Sabha and Spell Bee Olympiad exams.
Smart class for effective and innovative learning and well-furnished library.
Solitary importance on sports and games in state and national level.
WhatsApp facilities to know activities of students and school inside the campus.
Free bus for LKG to 8th standard.
Free, hygienic and nutritious food.
Neet, JEE, IIT coaching experts from Rajasthan and Andhra Pradesh along with our effective teachers.
Admission registration for classes LKG to +2.
KURINJI Education Institution, Kavetipatti, Namakkal. Admission in progress. CBSE, KG to 12, contact 9025895176. Metric 6 to 12, contact 9344567484. For online admission, log on to www.kurinjischoolnkl.in.
Emma, now in this video we are discussing vegetative propagation in plants, second video. So that's why here I have given 2. Vegetative propagation in plants 2. Okay, means which indicates video 2. Sorry, now let us see one by one. Now, previously we have seen vegetative propagation of stem. We have seen now we are observing the propagation by leaf, root, and bulbils. Likewise, there are different types of reproductive structures. Now let us see. So how these plants will be propagated, or how these plants are propagating. So by reproductive structures or vegetative propagules. Now let us see one by one. So right, now propagation by leaf. Foliar leaf buds or foliar buds are produced on the leaf margins of many plants. So means once you see there, so what are the buds which are present in the leaves or which are present at the leaf margins. So simply we will be taking them buds, or take that buds as foliar buds. Foliar buds are produced on the leaf margins of many plants. Okay, so these kind of buds definitely we will be taking as a foliar buds. Okay, now here what is happening means, so which can grow into the new plants. So these plants can grow as new plants. When such leaves fall on the ground, the buds germinate and gives rise to new plants. One more time, once you see there, what is the point? Leaf foliar buds are there. All these buds are produced on the leaf margins of many plants, which can grow into the new plants. When such leaves fall down on the ground, the buds germinate and gives rise to new plants. My point you understood. So easy English only I have used. So thereby, so what is the point here? We can understand some kind of foliar buds are there. Some kind of buds are there, or foliar buds are there, which are present at the leaf margins. Okay, so these leaf margins so consist of foliar buds. Now these foliar leaves definitely gives rise to new plants. When suppose if the entire leaf, when it is fall down on the ground, when it is fall on the ground, at the time what are the buds present on the leaf margins of that particular leaf definitely germinate and gives rise to new plants. So this kind of example, so where we can be seen means, we can be seen in Bryophyllum, and one more Begonia, and one more is Scilla indica. Bryophyllum I have written, KURINJI I have written, along with these two. So Scilla indica you can take, and one more is Begonia. Begonia also try to save in your mind. Now how many examples? Once you try to say along with me, Bryophyllum, KURINJI, Begonia, and one more is Scilla indica. Scilla, Scilla indica. So likewise, so there in a different types of plants, even leaves also helps in vegetative propagation. So means for the propagation of the plants, here the leaves involvement also is there. Leaves involvement also definitely is there, right? Now let us see propagation by root. So through roots also definitely vegetative propagation takes place. Now let us see somewhat examples. So now there tap roots of some plants, taproots of some plants develop adventitious buds. You see there how much clearly they have given. Taproot system is there. The taproot system of some plant develops what? Adventitious buds. Adventitious buds to form new plants. So these adventitious buds only definitely involves in the formation of new plants. Okay. So best example is Dalbergia latifolia. In a Dalbergia, suppose here if you see there, no, in Dalbergia definitely adventitious roots means by the formation of adventitious roots we can see the which one vegetative propagation. Next one, in some plants like sweet potato and dahlia. Sweet potato we know very well. Sweet potato Ipomea scientific name, Ipomea batatas, and dahlia it is a scientific name only, dahlia, DAHLIA, dahlia. There root tubers develop adventitious buds which develops into new plant. So same information. Okay. So root tubers here we will observe which one we will observe, root tubers and dahlia we will observe there. No, there which one root tubers. Tubers itself develops the adventitious buds. Don't forget one point here. If you take in a Dalbergia latifolia, taproot system is there. That taproot, on the taproot system we can find out the presence of adventitious buds. Whereas here in the sweet potato, and one more is dahlia, root tubers develop adventitious buds. Root tubers develop adventitious buds which develops into new plant. So thereby here what is the point here we can save in our mind means, in a Dalbergia latifolia on the taproot system, here we will observe the development of adventitious buds. Okay. Next one, on the sweet, suppose if you take some other plants here, on the sweet potato and dahlia, on the root tubers adventitious buds development is there. So thereby here what is the point now? So here also, so thereby here propagation by root. 2 or 3 different types of examples in a Dalbergia latifolia. Adventitious buds are there on the taproot system. Next one, in sweet potato and dahlia, adventitious buds are there on the root tubers. Okay, so this particular point you try to save in your mind. So means the position or the place of the adventitious roots or adventitious buds is different, but definitely helps in vegetative propagation. Definitely helps in vegetative propagation. So likewise also we can observe the development of or vegetative propagation simply, development of new plants. Okay, right. Next one, third one let us see, that is propagation of bulbil. How this propagation of bulbil is nothing but it is also involves, or bulbil is also some kind of important structure which helps in vegetative propagation. Actually this particular thing we will study under aerial stem modifications, but here in some other books they have given, they have categorized this particular thing as a separate. That's why I have giving this particular information separately. Okay. Now globobulbifera. In globobulbifera, some flowers in the lower part of the inflorescence are modified into small multicellular structures known as bulbils. In globobulbifera, some flowers in the lower part. Some flowers in the lower part of the inflorescence. So suppose if you take one inflorescence is nothing but a bunch of flowers or the group of flowers together and form one inflorescence there. So the lower flowers means the lower part of the flowers or the lower. So here some flowers which are present in the lower part of the inflorescence are the modified into how small multicellular structures. So these kind of structures only we will be calling as bulbils. So thereby they fall on the ground and grow into the new plants. Ipu paringa, some kind of bulbils development will be there. So when these bulbils fall on the ground, when they fall on the ground and grow into new plants. Okay, when these multicellular structures we will be calling as bulbil, when these bulbils fall on the ground, when these bulbils fall on the ground, so definitely these bulbils gives rise to new plants. Gives rise to new plant. Ipu bulbils means that is also one lemma. Adi small axillary buds we will study now. Axillary bud or terminal buds like these bulbils also some kind of bud like structures but helps in vegetative propagation. Actually here axillary buds we will not see at the inflorescences. No, that's why what are the buds present in the inflorescence or nearby inflorescence? So scientists they have given another name bulbils. These bulbils definitely are developed by the vegetative, either vegetative buds or floral buds. This particular point I didn't write, but through my voice you can get. So these kind of bulbils development we can see either vegetative buds or floral buds. So next once continuation you see there in American aloe vera. In American aloe vera, in American aloe, scientific name is Agave cisalana. In Agave cisalana, or you can remember that is American aloe or simply agave. Okay, cisalana, suppose if you have capability you can remember, no problem. Right, now agave cisalana, reproduce buds are nothing but bulbils only, often take place of many flowers on the inflorescence axis. Means here same thing. So what we have seen in Globa bulbifera, so there also the inflorescence what are the flowers are present there in inflorescence. Basal flowers all are modified into multicellular structures. Likewise in American aloe vera also means name is Agave cisalana. Reproduce buds are there. Reproduce buds means vegetative propagation buds only. So we will be taking them as bulbils. Often take the place of many flowers on the inflorescence axis. Okay, so on the inflorescence axis only, so these kind of reproductive buds or bulbils development will be there. These bulbils are also produced in the leaf axils. So here up to here two examples. One is Globa bulbifera and one more is agave. One is Globa bulbifera and one more is agave there. So what is the thing we are observing? The development of the bulbils we are observing where at inflorescences. So thereby here on the inflorescences also here we can see the development, or here we can see the development of bulbils which are helps in or which helps in which helps in where? Which helps in vegetative propagation. Okay, so thereby here one more important point, what is that? Not only these two, and here bulbils are developed or bulbils are produced in the leaf axillary position, or axils of leaf simply. So in axils of the leaf also we can see the development of bulbils. We can see the development of bulbils. So there here bulbils are developed at the leaf axils. So axils of wild yam. So here wild yam, likewise they have given one name. So I have given now wild yam scientific name is Dioscorea bulbifera. Wild yam scientific name is Dioscorea bulbifera. In Dioscorea bulbifera, bulbils are there at leaf axillary region. Leaf is there means in leaf axillary position, axillary bud will be there. No, that axillary bud only. Now here we will be considering as bulbil which helps in vegetative propagation. Point you understood. So thereby now what is the point? So here I told you already. Bulbils are developed from the vegetative buds or the floral buds. I told you. Now vegetative bud here, which one? Axillary bud we can be seen in Dioscorea bulbifera and one more is Lilium bulbifera. Next one, inflorescence buds means reproductive buds means where we can see or means here like a floral buds. Floral buds, this point you try to say. Floral buds we are observing in Globa bulbifera and one more is agave. Globa bulbifera and one more is agave americana. In some other books they have given, and in some other books they have given as agave cisalana, or simply you can remember agave and in a globa bulbifera floral parts. Means floral parts means which are present in the inflorescence develops into bulbils. Likewise in a Dioscorea bulbifera and in a lilium, axillary buds means axillary buds which are develops into bulbils. So simply here 3 examples are there. In 2 examples, bulbils or development we have observed in inflorescence. In 1 example, bulbils development we are observing in leaf axillary regions. Best example is Dioscorea bulbifera, and one more example, Lilium bulbifera. These are all we can be taken under natural methods. Vegetative propagation only, vegetative propagation in plants only, but through natural methods. Now let us have a look on the artificial methods now. Artificial methods also let us see. So there, first one let us take in artificial methods, first one layering. In artificial methods, the first one is layering. So there, in this method of inducing of root formation in the stem, while it is still attached to the parental plant, very easily we can understand. So means in layering, what we will do, just we will induce the roots formation. Just we will do what we will do, we will induce the formation of roots on the stem when that particular stem is attached to the parental plant. So thereby, so what we can do means here there are different types are there, but mother statement now let us see by one more time. What is that point? So in a layering, inducing the roots of formation means we will induce the roots of formation when, or we will induce the roots of formation in a stem when that particular stem still attached to the parental plant. So we can understand very clearly. So here this particular layering, for our convenience to study, so they have been divided into different types. Now let us see one by one. Right, now first one is tip layering. The sentence itself or the word itself giving information. Tip layering means, here layering means now only we seen which one, development of roots or the inducing of roots on the stem when that particular stem still attached to the parental plant. Ipu tip layering is nothing but initiation or the development of roots when that particular stem is present at the mother axis or the mother plant tip region. Okay, now let us see that only tip layering is nothing but tip portion of the is bent and buried in the soil. Tip portion suppose if you take a shoot system, definitely shoot system tip which one will be there, like apical root, apical shoot system is there, no? Apical bud will be there. Now correct word, apical bud or terminal bud will be there. So that particular tip portion of the shoot we are keeping, means we are bending and we are keeping in the soil, that is buried. We are keeping in the soil. Suppose one plant is there, that plant is standing aerially, that one now we are bending into the soil and we are keeping in the we are bending and keeping in the soil. Okay, so thereby what will be happen? So thereby here the what will happen means, so what is happening means definitely, so we can see the development of roots. So thereby tip layering best example is, once you see there, black raspberry. Black raspberry. One name black raspberry, best example of tip layering. So just a simple thing, tip layering. So what is the tip portion of the shoot is there, no? That one we are bending and we are keeping in the soil. So their best example is black raspberry. Okay, next one, serpentine layering. Okay, serpentine layering. 1 minute. Now serpentine layering. So there long slender shoot. Long slender shoot is bent and laid down or laid to the ground and covered with soil at a short regular intervals so as to form many parts Best example, clematis. Suppose when see here, once you see this particular statement, no, we cannot understand anything. That's why here I have drawn small rough diagrams. So based on the diagrams, let us have a look on these particular statements one by one. இப்போ பாருங்க, இங்க tip layering is there, no? Amma, இப்போ see there, these are all shoot tips, no, shoot system. So when this particular shoot system we are bending and we kept in the soil. So thereby when that particular shoot system touched to the soil, no, from there we are observing the formation of roots. That's why ipo pare, so roots development is there at the tip of the shoot system. That's why this particular layer type of layering we will be taking as tip layering. Next one, serpentine layering. Inge what we are taking, long slender shoot we are taking, and that one we are bending and laid to the ground, or laid in the ground, means we are keeping in the ground and covered with soil. Again this particular entire thing we are covering with the soil and we are keeping for short time. Short intervals when we are keeping, no, automatically what is happening, no. So what are the parts of the means what is the slender stem we have kept in the soil, no, their nodes and internodes will be there, no, at a nodal region. Definitely we can observe the at a nodal region. So definitely we can observe the development of roots. So thereby, so when we would like to separate, so there we can cut, so we can cut into mini parts, so we can use it we can use it for many purposes, means we can replant that particular plant in another place. This kind of phenomenon or this kind of type of layering we can be seen in clematis. Serpentine layering one illama, what is that, no, one long slender stem or one long slender shoot you take, ange konjam soil ellame pakkathile potti idu stem ange potti, again we have to keep that particular soil on that particular what is the slender branch we kept in the soil, you know, on that particular shoot, you know, we will be keeping soil. After a few days definitely roots formation takes place. So later that one we will cut and we will use it for another purpose, several purposes. Next one, best example in a serpentine layering, clematis. Next one, trench layering. Trench layering also will be like that only. Once you see their diagram is this with the white color chalk piece I have drawn, now that one, trench. Okay, now there again we are taking long shoot is placed in the trench. Again same like a dig, we will dig the soil and we will that is only trench. So long shoot is placed in the trench, leaving the apical portion is exposed. Here this particular apical portion must be exposed. So that particular apical portion must be exposed, but that particular short shoot must be there in the trench. This is soil level. Now this long slender shoot is buried in the soil, but here what is the precaution we are taking? Here the shoot systems should not be buried, that must be exposed in the air. This is soil level. So thereby the roots are produced at each node. Roots are produced at each node on the lower side, and the shoot system or shoot emerges on the upper side. So here from the upper side shoot system development, and from the lower side root system development we can see. This kind of a thing or this kind of phenomenon we will be taking as trench layering. Okay, trench layering. So there this kind of layering type we can be seen or we will observe in walnuts, and one more is mulberry. Walnut plants as well as in mulberry plants. So definitely we can see this kind of trench layering. Serpentine layering we can be seen in clematis, and trench layering we can be seen in walnuts and mulberry. Okay, next one, simple layering. Next one is simple layering. Rooting is induced on a soft stem. Rooting is induced on a soft stem, and it is defoliated and small injury is made on it. Means here what is the thing? No, definitely we can induce the roots on the stem. soft, on a soft stem. But what we are doing? First we are doing, what are the leaves are present on that particular shoot stem? No, we are removing. Which one we are removing? Leaves. That's why we have used one word, defoliated. D means removal. Which one we are removing? Leaves. That's why defoliated. Defoliated means a stem, and a small injury stem means wantedly we are keeping one small injury. Small small injury we are doing or keeping, or by keeping of that, by injuring of that particular soft stem, now we are keeping in the soil. Once you see there, one a soft stem we took, so there what are the leaves are there on that short stem, now we have removed and we kept in the soil. So thereby what happened, now let us see. After that it is pegged in the soil. Already we seen this one, to develop adventitious roots. So here the adventitious roots development we are observing in a diagram. Later on the layer is separated and planted. So here we can be separated. Once you see then, so this kind of thing, so we have seen in stolons, no. Again one is a particular that grows obliquely downwards. So likewise we can be seen in jasminum or jasmine, and one more is grape vine. Grape vine and grapes plants. Okay, grape vine is a scientific name. Vitis vinifera, jasminum vinifera, jasminum da. Right. Now simple layering jasmine, and one more is grape vine. And one illa, what we are doing, just we are taking one soft stem and simply wantedly, so we are removing all leaves, and a small injury we are keeping, or we are giving to that, and we are burying. Means here we are keeping in the soil. So thereby after some time, no, definitely roots formation takes place. So this kind of a phenomenon exclusively we can see in jasmine, and one more is grape vine. Next one, air layering. Air layering or gooting. This is important. Air layering or gooting. Air layering, the word itself giving, we are not keeping this particular layering in the soil, we are trying in air. So that's why scientists, they have given a name as air layering. We are calling this particular air layering by one more name is gooting. Now rooting is induced in aerial hard branches. Rooting is induced in aerial hard branches. The stem is girdled na mele stem mele ipo brown color layer phloem present, no, that particular thing we will remove. Ipo girdled means mele, suppose here in a secondary growth, after in anatomy we have learned this particular point. Girdled, suppose if the stem is girdled, in a transport in plants we have learned, suppose if the stem is girdled, so at the time what is a part of the plant dies first. Likewise we have learned one important bit now there. So likewise here also what is the thing we are doing, the stem is girdled means outer protecting layer we will remove. After removal of a protecting layer, white color thing we will observe now. So that is the thing, girdled and covered with moist moss or cotton. And where we have removed outer layer, no, there white color part we will observe, no, there what we are doing, that particular part we are covering with moist moss or cotton. Okay, so later water is added to it along with a small quantity of root promoting hormones. Means here water we are adding, so we will add water to it. Later somewhat very small quantity of root promoting hormones also we will add to that. Ipo once you paringa white with white color chalk piece chinna box madri is there, no. So there what we added water and small quantities of root promoting hormone also we added. Ipo with orange letters small coils are there, no, adenna rope chinna thread like structure mele pot I have drawn. From this pot to water tanni kile varo. So idellame all the time will be there in wet condition, moist condition. To maintain the moist condition, here one pot I hang panni. So from this pot to we have taken water supply through the rope. So thereby what is happening, after 2 to 3 months roots appear and shoot system is then cut below the cotton bandage. Ipo after 2 or 3 months, 2 to 3 months, definitely roots we can appear, we can observe here. So thereby here we can cut this particular plant or particular branch or a particular shoot where exactly at spotlight, means exactly below this or below the cotton bandage. So below this one here we can cut. Below the cotton bandage and replanted. Why? Because definitely here the roots and everything is there now. This particular branch we can be replanted. So best example is lychee and one more is pomegranate. Pomegranate and one more is lychee. This kind of in lychee and pomegranate, no, this kind of air layering or gutty method we can follow. So here the mass or the cotton bandage. So here and from here to orange orange diagram like coils thing is there, no. Again from here from one pot we are supplying tanni water. So these are all rough diagrams of different types of layerings. So in the next video I will continue to give information regarding artificial methods. So this is the second video on Neet classes. Topic name is vegetative propagation in plants. Topic name are not required, but what is the information till now I have given explanation, that you don't forget please. Try to watch this video once or twice, so thereby you will familiar. If you get the knowledge means you will be benefited with good marks. Take care and be safe at home.